Because my ultimate goal is to start with a fairly boring plant like Petunia, and then modify it such that if I put it in front of a panel of like world-class botanists, they don't know where to taxonomically key this out. They don't know how to define this on the tree of life.
Hey guys, today I'm interviewing Sebastian Cocioba, who is a self-taught amateur biologist who genetically modifies his plants using some very cool technology and protocols that we're going to talk about today. I'm going to have all of Sebastian's links below in case you want to dive deeper into his work and maybe even eventually start genetically modifying your own plants. Let's get started. Hey Sebastian, so first of all, thank you so much for agreeing to come on the channel. I've been watching content that you've done, whether it's interviews or the Thought Emporium video, so I'm pretty familiar with your work, but I'm really excited to share it with my YouTube channel today. If you could give kind of an introduction of who you are and how you got started doing what you currently do, that would be awesome.
Sure. So first of all, thanks for having me. So my name's Sebastian Cocioba. I'm a amateur biologist, I guess. I have no formal training. I hold no formal degree. I work out of my home lab, which I've been doing for the last 15 years as an contract research organization, kind of like a one-man pipette for hire. That's how I pay the bills. And then in my spare time, I'm aspiring to become a flower designer. So the goal is to make like Dr. Seuss level flowers out of what we see in front of us using genetic engineering.
I first became aware of your work from an article on chemistryworld.com, and it mentioned that you had purchased a gene gun from China. And as soon as I read that, I was like, I have to interview this guy. Like, that is crazy. Would you mind kind of giving us an overview of flowers for everyone?
Yeah, sure. So where to begin? Okay. So again, like I said before, I've been doing this for like 15 years, and it's been really fun. Very, 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 very difficult, but very fun. But one of the most important things that I've learned is that it's pretty lonely doing it by yourself, right? And you hear a lot within the amateur biology circles of DIY bio, right? Do-it-yourself biology. And I got a little tired of doing it myself, and I'm looking for like do-it-together biology. Because one of the issues is that having the geographic privilege of being in a country that doesn't regulate biosafety level 1, and having access to these materials and the space to do so. And even though it took me 15 years, I still have a pretty robust laboratory with a lot of fancy stuff that if you'd buy new, it'd be very, very expensive, right? So like I taught myself electrical engineering a little bit to learn how to fix and flip equipment on eBay, kept the best stuff for myself. So it took a long time. But looking back, the 15 years really took, it took too long, right? And like, I think now with hindsight 2020, but I think with good mentorship, people can get up to the same level where I am fairly quickly, like maybe within 2 or 3 years, right? And so I kind of embarked on that journey of how can I pay it forward and kind of bring people into the fold, right? Like grow friends, right? And so I started this group called the Petalsmiths Research Party on Facebook, back when Facebook was not cool, but at least popular.
Yeah.
And it was really, really fun. And I started noticing that the bulk of the people who were interested were from other countries, right? And so I tried to help them within the letter of the law in their country, because certain countries just like ban genetic modification whatsoever, right? But they still wanted to do science. So I wanted to figure out how can I, without like, undermining their local laws, because I don't want to get them in trouble, how can I assist them in their journey in any capacity, right? Be it plant breeding or tissue culture, hybridization, stuff like that. And I started noticing that even in a school setting, things like antibiotics are very, very regulated and hard to get your hands on, right? And so I went to a really crappy high school when I was a kid. And we barely had any budget. Like we didn't do any type of laboratory exercises. It was all like chalk and talk. Right. But like 13 blocks away, Dalton Preparatory School, which is this like $6,000 a year high school, we're doing yeast bioengineering day one. Right. And so I felt that like the only way for you to have a good biotech background is to have the privilege of having 60K disposable income. It's not that you, the student, let's be real, the child is like more gifted or more intelligent than their peers. It's just that you don't have access to resources. So resources became like a huge narrative for me, trying to figure out how can I lower the barrier to entry. And And what I noticed were like two things. One that you can definitely relate to in terms of the distribution of knowledge with things like tissue culture, right? How like the knowledge is out there, but it's very disparate and like distributed across many hyper-specific texts.
Yeah,
And
and you almost have to, like, put it together, like, take a little bit from here, like, that's how I initially figured out how to do it, but it was
same.
hard.
Yeah,
Yeah.
yeah, same. And if it wasn't for the book Plants from Test Tubes by Holly Scoggins, I got it in its first edition way back. Now it's in its fourth edition. And that was my Bible growing up, right? Like, yeah, there you go.
My laptop
Beautiful.
stand right now.
Yeah. Yeah. So that was such a seminal book for me. And I'm sure for you as well. And the core thing is that that was a compressed tome of knowledge that a technician level understanding of plants would be enough for you to work with that space. And so I started this project called Flowers for Everyone, which kind of self-explanatory. I just want to make flower design accessible to as many people as possible. And the first thing was like, okay, getting your hands on protocols and techniques and things necessary. So on our Binomica Labs GitHub, we have this like 100-page picture book of how to do molecular cloning, from pulling a gene into a plant to putting in bacteria to sequencing it. And then chapter 2, which I want to write soon, would be putting that gene in the plant, right? So like being able to like interface with the organism, right? And along that way, I started like listing what were the most limiting factors of this process. And the two major things were antibiotics and herbicides, right? And in antibiotics, especially for molecular cloning, you need to do that in order for you to construct plasmids and then select. Plasmids are little circles of DNA, kind of like scripts, that you put in bacteria. And a special kind of bacteria, Agrobacterium tumefaciens, transfers that DNA into the plant, right? So you construct your genetic circuits in E. coli, and then you do so by selecting for the cells that don't carry your plasmid using antibiotics. When you move the plasmid from E. coli into Agrobacterium, you also have to select away the Agrobacterium that doesn't have your plasmid, and you do so as well with antibiotics. And the idea there is that along with your gene of interest, your genetic circuit that you want to put in the plant, let's say make it glow in the dark or turn blue, right next to it is the gene for antibiotic resistance, right? And that'll allow the bacteria that carries your genetic payload to be selected against all the other bacteria that don't have you, right? And so some antibiotics play a massive role in the construction of genetic elements. And now on the plant side, whenever we transfer DNA from the Agrobacterium to the plant cells in tissue culture, the cells that don't carry your gene of interest need to be killed off in order for you to regenerate a whole plant that has nothing but your gene of interest and no wild type versions. And so to do that, we use the antibiotic equivalent in plants called herbicides. And so these herbicides naturally kill the plant that you're working with, but if it has the resistance gene, it doesn't. So the only thing that grows on your media that has herbicide in it is your plant with your transgene inside. And so these two chemicals, these antibiotics and these herbicides, are like absolutely critical in the process of constructing genetic circuits and then putting them into a plant. And so I was kind of hitting my head against the wall trying to figure out how do we go about this without antibiotics, right? And so starting with the plant side, there's this really cool patent out of Syngenta. Syngenta is this really big plant company, breeding and like seeds and chemicals. And they came out with a patent that says that they realized that plants cannot metabolize mannose, right? Mannose is this sugar that you can buy over the counter. It's used for urinary tract infection treatment, right?
Mhm.
It's a super safe, just regular sugar. It's just a particular kind of sugar. And so plants can't eat that. Now, there's a gene in E. coli that allows E. coli to eat mannose. And what they saw is that if you take that gene from E. coli and put it into a plant, that plant can now metabolize mannose. And so if you make a tissue culture media where the only carbon source instead of sucrose is mannose, the only thing that will survive are the cells of plants that are transgenic, right? So you're doing positive selection, but without the need for herbicide. And we're not transferring the herbicide tolerance genes, which is always a problem, because weeds can pick up pollen from other plants and then they become super weeds. Because along with your cool gene for glowing, you also have herbicide tolerance that's really important in agriculture to protect weeds against our to protect our crops against weeds. So the more we limit the use of herbicides, the better it would be. And as amateurs, we definitely want to be environmental stewards. So we definitely don't want to work with genes that spread the tolerance of herbicides, right? Especially because these chemicals are really regulated. So now we have the ability to select plants with sugar. Now, taking that same inspiration, I realized that the workhorse E. coli that we work with in laboratories, the super safe, non-pathogenic E. coli that we use to construct our genetic elements inside, happen to not be able to eat sucrose, table sugar, right? So the wild type pathogenic nasty E. coli can, but ours can't. And so I took the gene digitally from a sequence from the wild type E. coli, and I put it on I put the gene for sucrose metabolism, the CSCA gene. It's an invertase that cuts sucrose into glucose and fructose. And then that gets metabolized further. So I took that gene, I put it on a plasmid, and then I made minimal media similar to like plant media, where the carbon source, instead of it being glucose, was sucrose. And now all the cells that don't carry my plasmid can't grow because they can't eat. And the ones that do can, the ones that can eat the sucrose will grow. And so now you have positive selection using sugar instead of antibiotics, right? So slowly but surely, I'm cobbling together an end-to-end pipeline where you can build genetic circuits in E. coli with sucrose, and then transfer those into Agrobacterium, and then transfer those genes from Agrobacterium into the plant using nanos. And so it's antibiotic-free, herbicide-free, and essentially less of a regulatory burden, such that other countries could potentially make a plea for, at least in the high school setting, to be able to do genetic engineering and expose the students early on.
Can you modify any trait of a plant, or are there specific traits that are really hard to modify? And what specific traits are you specifically focusing on?
Yeah, for sure. Great question, by the way. So DNA is DNA is DNA. And so as a result, we now have tools like CRISPR that can modify the base pairs of genes that are already present. And we can do so with pretty high precision, such that essentially any gene within the plant is fair game. The hard part is actually knowing that the gene is there and knowing what that gene is, right? So like the model, we use the word gene as like an individual unit of function, right? Like a trait, right? And that gene could be a cluster of multiple coding sequences, right? That encode different proteins that come together to make that one functional thing. When the more advanced of a scientist you ask what a gene is, the more elaborate of a question it becomes, right? Of like, what is a gene? Like, what is a gene? Where do we define it, right? But not to get in the weeds and sidetrack, but essentially, what I'm interested in are body plan traits, right? So these are genes. They're called homeotic genes, and they're responsible for the systematic construction of a plant over time, right? And specifically, I'm interested in floral developmental homeotic genes, which means the genes that are responsible for the elaboration of petals, for the formation of stamen, of stigma, of styles, et cetera, et cetera. And so I'm a big fan of petals. I love petals. I think that they're nature's canvas. And one of the reasons why I'm so interested in it is because they're a vehicle in which the plant gets to express itself. And it's not hyper necessary for its individual survival, but it's necessary for the survival of its species. So it allows for tissue that is kind of like a living canvas that you can play with without messing with the actual underlying engine of the plant, right? So it's like this one area is a sandbox that you can do essentially whatever you want without impacting the survival of the organism, because you are the caretaker of it at this point, right? It doesn't need to be open air pollinated. And if it's a transgenic, you absolutely can't do that, right? So anything transgenic can't leave my laboratory. But within the confines of my lab, I'm the selection pressure, right? So like I choose which one I want to keep, and my main interest is in shape. So there are certain genes that get turned on at very particular points during the development of a plant, of a flower, that then beget its shape. Right. And there's like a whole network of genes that repress those genes and activate those genes, and other proteins that come into play. There's now microRNA which throws another wrench into the equation. Really, really tiny sequences of RNA that bind to these proteins that interfere or activate their ability. And so all of this comes together in this like beautiful orchestra that then turns into a flower, right, over time.
Mhm.
And so by tuning and tweaking those, I can then change the shape and the structure of those petals. Because my ultimate goal is to start with a fairly boring plant like Petunia, and then modify it such that if I put it in front of a panel of like world-class botanists, they don't know where to taxonomically key this out. They don't know how to define this on the tree of life
Mhm.
phenotypically, right? So just by aesthetics alone, I want it to be a flower no one's seen before. But more importantly, have the pipeline so students, kids, people of all ages can design their own flowers.
In terms of the methods that you're using to change these flowers, is it mainly the agro-infiltration of the Agrobacterium, or is it I guess I also have another question. Sorry, I ask multiple questions at once all
Please.
the time, and it's a problem. But there's that piece, and then I'm kind of confused how the gene gun fits into the genetic modification, like if it gets used in tandem with the Agrobacterium or if it's an entirely separate process.
Sure. Okay. Let me start with the second question, and then that'll feed into the first. The first is a
Okay.
little bit more elaborate. Okay. So gene guns. So gene guns are essentially genetic shotguns, right? And the idea is that if for some reason agrobacterium doesn't agrobacterium based transformation of your cell of your tissue doesn't work, right? Like agrobacterium acts like a shark in the water, where it just kind of does its soil bacteria thing until it smells plant blood. And then it kind of goes into a frenzy and activates genes inside of itself to make these little needles stick out all over it. Those needles is what the agrobacterium uses to transfer its DNA from itself into the wounded plant tissue, right? In nature, if you've ever seen those giant tumors on trees, like those big old growths, like in
Yeah.
a park, yeah, that's mostly agrobacterium, right? That's in the rhizobium family. And these are bacteria that transfer tumor causing genes, essentially auxin and cytokinin mass production genes, right? And such, it basically makes a callus on the plant, right? And along with that callus, it also encodes a gene for starch that it can eat, right? Only the bacteria can eat. So it makes a tumor and feeds itself by genetically engineering a wounded piece of the plant. Yeah. And so, a scientist in 1979, Mary-Dell Chilton specifically, she figured out that that's the case, right? That the bacteria is actually genetically engineering the plant and not just secreting hormones on the surface, which was a huge discovery that required a ton of work to really prove, because everyone was not believing her. She was also a woman in science, so there's just like disbelief on top of prejudice, on top of dogma, and so she did heroic effort and proved the unimaginable, that nature does genetic engineering, that it's possible to do this. And so she then went on to modify that bacteria and remove the tumor causing genes and replaced it with a little placeholder that genetic engineers add genes to that area. So instead of making the tumor genes transfer into the plant, it makes whatever you'd like. It put whatever genes you'd like. So like human insulin or the genes for glowing in the dark or making it blue. And so that whole process relies on plant bleeding good, right? So like if the plant produces certain phenolic compounds, these essentially plant blood. So like the
Mhm.
smell of fresh cut grass is just phenolics. It's plants screaming, which is kind of dark. Um,
Oh my god.
so so yeah, that's why like mowing the lawn after doing all of this just makes me feel bad. And so that blood is super important, because this Agrobacterium tumefaciens, the bacteria itself, evolved to sense that, to know when to turn on these really genetically expensive genes to make those little stabby bits, right? And so if the plant you're trying to work with isn't within the menu, the repertoire of Agrobacterium's usual suspects, you need to use synthetic plant blood. There's a chemical called acetylsyringone that kind of sorta it mimics plant blood and it activates those genes, but because of the structure and the cuticle and all the other anatomical bits of certain plants, it might be too difficult or near impossible for the agrobacterium to properly bite down and transfer those genes in, right? And so people have been looking for a long time for alternatives to agrobacterium because of this limitation. And so enter the gene gun. So like I said before, what a gene gun is a genetic shotgun where you take your DNA and you coat it onto these little nanoparticles of gold or tungsten. I use tungsten. I'm not a rich man. I also don't feel sane shooting gold and then throwing it down the drain. That's nuts. So I have these tungsten nanoparticles. They're about 800 nanometers wide, right? Um, and I coat it with the plasmid, and I use this like extract from salmon sperm called spermidine, which I know it's super weird. But it's a chemical that allows for DNA to glue onto things, right? And evolutionarily, that makes sense because you want the sperm of salmon to bind to the eggs of salmon, where all of this is happening outside of both of the salmon's bodies, right? So salmon lay eggs, and then the female salmon lay eggs, and then the male salmon kind of crop dusts over it with its sperm. And so nature evolved a really sticky genetic material that allows for like the binding specific. It's like DNA glue, right? And so you glue the DNA onto the tungsten particles with salmon sperm extract. You let it dry onto this little sheet of plastic, and the sheet of plastic acts like a shotgun shell. You load it into this cylinder of the gene gun, which looks like an espresso machine. And there's a really short runway of about like 2 inches that under high pressure, that little disc gets accelerated onto this mesh at the very bottom.
Mhm.
So when it smacks that mesh, the particles then continue, because it's in a vacuum, it continues down at the acceleration, at the speed of the disc onto the tissue. So it literally shoots the particles into the cells. And then through black magic that we kind of don't really understand, the DNA gets integrated into the plant genome. Right?
Wow.
Yeah. And so you're basically causing a stress shock wave that upregulates the machinery for genetic repair. And then you're just like cramming bits of DNA into the cell, ideally in the nucleus, maybe, where then the DNA gets integrated basically randomly. Right. And so this is a shotgun approach. It's really not elegant. And as a result, it's like 2 orders of magnitude less efficient than Agrobacterium. An Agrobacterium run, if you're using a model organism that agro likes to bite on, like tobacco, for example, I get efficiencies of 85 to 95%, right? So 85 to 95% of all the leaf discs that I hole punch and expose to agro will produce a transgenic plant. While with tissue culture, with a gene gun, we get about like 0.1% efficiency. So you really have to shoot a lot. But at the same time, every single tungsten particle has your plasmid, and every impact event will be another chance. So you basically group them all together on a petri dish, real tight around a center that you've predefined by shooting onto paper first and see the little splatter. And then you just blast it, and then you transfer it onto media, and you nurture that through tissue culture. Then after like about a week, you then transfer the explants from happy media to media with herbicides to start killing off the non-transgenic tissue.
Just to clarify, with the gene gun, you're shooting it directly into like a petri dish with your leaf discs, or you're shooting it directly onto a existing plant?
So you can do both, right? So there are 2 models of gene guns. There's the low pressure Helios version. So this company Biorad came up with 2 different gene gun models, a desktop and a handheld gun model. The desktop model can do really high pressures and can penetrate deeper into the tissue, while the handheld model is more of like a transient version of the other gene gun, where you shoot it in, it'll do its thing, but it didn't penetrate deep enough to get to the nucleus. At least that's the working idea. The one I have is the desktop model. So I put explants leaf discs into a petri dish, and I just blast them directly onto it. And I do about 10 shots per experiment. So 10 petri dishes of like, let's say 20 leaf discs in as tightly packed array as possible.
Mm.
But the nice part about the gene gun is that it's kind of species agnostic. Like it doesn't care what you put in front of it. In fact, gene guns have been used to transform mammalian tissue, bacteria, algae, archaea, anything, because it's physical. It's not limited by the genetic machinery or chemistry. It's literally just shoot particle into tissue, right? And for a while, people were considering, and there has been, if I remember correctly, there has been actual clinical trials of using the handheld version of gene guns as genetic therapy in humans, of just like shooting cancer with a specific area, shooting and silencing genes that break the machinery of the cancer cells. So it doesn't care what you put in front of it. And as a result, you can transform like literally any plant. But the problem there, it's actually not in the gene gun, it's in the tissue culture. So everything that I've described to you thus far about transgenic plants, be it with Agrobacterium or with gene guns, specifically require you to have a robust tissue culture protocol, right? The more efficient your regeneration protocol is, the more effective your transgenic pipeline will be.
I have
Like,
such a dumb question, but I think
please.
hopefully I'm not the only one. But what is a transgenic plant?
Sure. Okay, so there, this is a good little vocabulary lesson. So there are two types of genetic modifications. There are cisgenic and transgenic. So cisgenic means you take genes from inside the plant and you move them around to do something new, right? So like if there's a really powerful promoter, the thing that drives gene expression, driving some other gene, and you want it to overdrive the genes for color to make like an apple that's red everywhere, you can take the promoter from that one gene and then the gene for red in the apple and put them together and put them back in the plant, right? So that's using its the natural gene pool's genetics, but rearrange it in a different context, like taking
Okay.
words from a sentence in a book to make a new sentence,
Hmm.
right? But it's still from the same book. Now, transgenics is when you take genes from other organisms outside of its gene pool, right? So, like if I were to take a gene from salmon and put it in a tomato, that would be a transgenic tomato. And
Okay.
more often than not, they say it's uh, specifically foreign DNA, not from within itself.
Okay. So, for example, like the LightBio glow in the dark Petunia would have been a transgenic plant.
Very transgenic, yeah, because
Okay.
it not only uses genes from other plants, but it also uses genes from fungi.
I did a video about that specific plant a few months ago because I got mine early. And it was before I had started kind of researching the Agrobacterium, and I wish I could redo that video because I feel like I understand it so much better. But in that video, I basically got it in the mail and I was like, yep, it sort of glows. Cool.
So I was in the lab with in Vitaly Sitovsky's lab at Stony Brook University back in 2010, when they were making their the very very first glowing plant before LightBio, before the Kickstarter glowing plant. This was the very very first one, and they used the gene gun and they transferred the genes from this bioluminescent marine microbe into the chloroplast of a plant, right? Which is even harder and even lower of an efficiency because you have to go through the cell wall, the cell membrane, and then the membrane of the chloroplast with a single tungsten pellet, right? So really, really low chance of it working. But it did. And they managed to do it. And it was just this like little thing in a tissue culture vessel. And they made it, it was so poorly executed. They made it as a tissue culture product where they
Mhm.
would sell you the plant, but then they like glued a piece of plastic around the neck of the plant on top of the media to keep you from pulling the plant out of culture,
Oh my
and they made you like
God.
sign a thing that says you will not take it out of culture, you will not propagate it under penalty of law, blah 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 blah. And because it was in this magenta jar, right, with the sealed lid and no way to change the media, it was just dying in your hands, and so you paid
Mhm.
$64 to watch this thing like suffer and die, right? And when I saw that first glow, I'm like, yeah, okay, it was glowing, but like I was like, this is definitely the first, and it won't be the last, and it'll be better and better, right? Because I really was not impressed. And
Mhm.
then the light bio plant came, and it's cool. It glows, and it glows sustainably, right? It recycles that spent fuel, so that it's easy to work with and whatnot. But like, it's still, it wasn't more than just like, okay, that's really cool, you know. And congratulations to the team. That was a huge effort. But like for me personally, as someone who's like steeped in the lore of flower design, I'm just so not interested in glow, right? Like
Really?
I thought I would be, but just like the energetics around this is just so complex that the day we have something super bright will still just be this like bright flower in the night, and it won't really be possible. It'll never get brighter than a firefly's butt, for example, right? Like
Yeah.
unless we do something like really out there. To the best of my knowledge and my understanding, like continuous sustained glow brighter than a firefly's butt is probably physically impossible with what we know
Mhm.
so far. But like um seeing that in person actually cemented how much I care more about shape, right? In
Yeah.
my personal like art practice, I guess. Because I wear a biologist hat because I don't have the tools for doing the practice as an artist yet. And I hope to one day take off my biology hat and put on my artist hat, because it's like imagine being a painter, wanting to be a painter in a time where there was no pigments and you have to go to the mine and dig out some ore and grind it up, and like what is a canvas, how do I stretch this, like we're barely there, you know. So I'm a
Yeah.
little too early on that.
No, I think that's a great analogy. we can get off the LightBio stuff because I want to learn more about agroinfiltration. But
Sure.
the last thing I want to ask you just quick about the LightBio plant is, do you think there's issues with it being sold commercially and them being outside potentially and being pollinated with
No.
pollen from other petunias and things like that?
Yeah, no, I don't think so because like so it was deregulated legally by the USDA, right? And
Uh-huh.
they recently made more sane laws that focus more on the regulation of the mechanism of action, which basically means like what is your plant doing, right? Like what is it producing? What is it doing? What would those genes do if they were in other organisms? And the interesting part here is that it's so metabolically expensive to make all the proteins necessary to glow that it's like, imagine I give you $100 to survive the week, and halfway through the week you spend $50 on shiny pants, right? And now you have to make a decision. Either you give the pants back, silence the gene, right, and survive the week, or you suffer and maybe die, right? And so there's like this like budgetary decision, not decision, but this budgetary pressure put on these plants to be able to survive in the wild. And so
Mhm.
by adding these genes, you burden the plant more than its wild cousins that have been kind of like streamlined and optimized to do their thing to the best of their ability. And so this burden, more often than not, makes them less successful in the wild unless it unlocks a new like sexual niche. Like you're suddenly getting pollinators that you'd never get before, like
Mhm.
at night by ants or something like that. And so if that pollen goes from petunia to petunia, that trait will more than likely just vanish throughout the course of generations, like very quickly, because it doesn't have any evolutionary benefit, right? Like it's harder for it to do the same thing that the regular petunia does. On top of the fact that we don't eat petunias, they're not part of our food chain in any capacity. They're originally from like southern Peru, for example, and so they don't grow in the wild anywhere here. So one of the rules that I have for genetic engineering is thou shalt not edit weeds, right? Like don't do that, and don't edit things that are like super spready, right? Like you don't want something that you don't want to take a native plant from like your backyard and genetically engineer that because it already is adjusted to your local environment. So it'd be
Yep.
more than likely to spread. On top of the fact that it's a federal felony to not release a plant without deregulation, right? Like outside of the law, just purely on ethics stance. My take is always, what am I doing to the environment by giving this plant, by releasing this plant? If I were to release one that has been deregulated, I'm saying within that
Mhm.
hypothetical. And then the second part, because I know people would say like, oh, but how do you know, right? How do you know what would happen? And one of the questions, the true answer is you don't fully know what would happen, but you can take a very, very educated 80 years worth of research guess as to understanding the ecological distribution of novel genes within a population, right? And so there are ways to actually model this. There's a lot of research being done on upon deregulation, what does that do to ecosystems? And we've had many transgenic plants in the wild. Not many, but a lot of hectares of our farmland is mostly transgenic plants. And what we've noticed is that um, we have some pretty good data on showing like how it doesn't spread, right? And if those genes if the plants take up those genes, they're not really going to use them in the way that we want to, right? So yeah, long story short, there's a lot of reasons as to why upon deregulation, there's not really an issue with them being out in the wild. It really depends on what you're trying to do. And for things within my sandbox of like make a flower that looks weird is probably going to be really off-putting to the natural pollinator anyway, right? So even if I'm just changing shape, it's still going to give us this evolutionary burden, right?
Yeah.
Against that. Yeah. Sorry for the long-winded answer. It's a
No,
super
it's
complicated topic.
fascinating. So I've seen a lot of photos of the agroinfiltration that you do, and you post a lot of content about it on Twitter. So I was wondering if you could walk through how the process of actually injecting the agrobacteria into the petals of the petunia flowers actually works.
Sure. So first to give a little bit of context, right? So there are two types of plant transformations. There's stable and transient, right? Stable transformations means that the gene is integrated into the plant genome and that you've through tissue culture reproduced only tissue that carries that gene, such that every single cell has your gene of interest, and that resulting gene is heritable. So if the plant makes seeds, the offspring will carry that gene as well. Transient, like the name implies, is that it's temporary, right? And so before I commit to like a multi-month long, let's say 4-month long campaign of transforming a petunia with a gene of interest, I want to make sure it works, right? And so what you can do is that you arm your agrobacterium with your plasmid through a process called heat shock, where you expose chemically treated cells, mostly with calcium, chemically treated cells with your plasmid. And then that plasmid, upon heating and cooling, will enter the membrane of the cells and then be trapped in there, right? So now you have your plasmid inside of Agrobacterium. So because of the mechanisms that's inside that plasmid, it'll also give the Agrobacterium the ability to survive on media with antibiotics, specific antibiotics. And so you plate the agro on media with antibiotics. I like, on top of the antibiotic gene, I like adding a visual marker. So all my Agrobacterium, when they're armed, they turn pink, like really, really bright pink. And so I can just look on a plate of white colonies and pink colonies and say, okay, those have my plasmid. No guessing, no PCR, no extra steps to verify. So now you have armed agro on a plate. You take a scoop of that and you put it in a tube of the same media with the same antibiotics and in liquid form, and you let it incubate overnight, about like 12 to 16 hours. And as the cells grow and divide, they make the media more cloudy. So you can either use a device called a spectrophotometer to measure how cloudy, or you can kind of eyeball it against a mental reference. I eyeball it because it really doesn't matter too much, especially with these type of temporary tests. Okay, so now we have Agrobacterium growing happily in a little tube. So I spin down the tube and I get rid of the media and I replace it with this media that is low pH and has that simulated plant blood inside, right? And now this begins the process of inducing virulence, of making the Agrobacterium go from little soil microbe to like really angry, spiky thing that wants to bite down on stuff. And so I found in this really awesome Indian paper that vanilla, cumin, and cinnamon all induce Agrobacterium to varying effects, as well as the synthetic plant blood that's really expensive, like $100 for 10 milligrams, versus vanilla that you can buy at a grocery store. So I use vanilla. And so I add a little bit of vanilla to that tube, and I incubate it in the dark for about 4 hours. And that's kind of a rule of thumb. I'm actually working on a genetic circuit so that the bacteria changes color again when they're fully virulent and angry. So I know exactly how long until like the water's chummed and they're really ferocious. Because you can't really tell. The only thing that changes is that the culture media odor goes from like poop bacteria to this just like wet cabbage, right? So from one unappealing smell to yet another one, but it's very different, right? The only saving grace is that the vanilla overpowers all of that. So every time I do a transient infiltration, my whole lab smells like a bakery, which is great. Yeah. So all right. So it stays in the dark, wrapped in aluminum foil at like not body temperature, but kind of warm, a little bit warmer than room temp for 4 hours. Right. And so now you have angry, spiky Agrobacterium. You load it into an oral syringe, which is a blunt syringe that has no needle, right? And it's kind of rounded. It's used for like babies and like small animals to inject food or medicine. Again, with no needle. And with gloved hands, you approach the pedal from above, and you push against the against the nose of the syringe on the other side, and gently squeeze the Agrobacterium into the pedal tissue. And so there are layers in the pedal that have that are kind of disconnected. And what you're doing is you're injecting through small micro wounds that are caused when you push the blunt nose of the syringe onto your finger, and it basically streams in between the layers all of this Agrobacterium, right? So it actually enters in between the cell layers and will kind of hang out there for a while. And so what's happening is that now you have this like spiky, angry Agrobacterium in direct contact with a whole bunch of these cells, right? And so these cells are going to be transformed, and the bacteria is going to transfer the DNA into it. But so many of those bacteria are constantly making the little genetic packages and putting them down their little tubes that the general bulk of all of the inserted DNA elements are going to express in the cell media, like inside of every cell. It's like a super loud temporary blast of genetic material inside all of these cells. And so that you get this like really massive signal of whatever you're trying to do. And so when I want to test the efficacy of a promoter, for example, how the thing that drives gene expression, I normally, instead of my gene of interest, I drive a green fluorescent protein, something that gives a color, or the ruby cassette, which produces beet juice, essentially. And how much green or how much red is present gives you an indication of how powerful that promoter is, right? And so you can rapidly prototype by injecting the agrobacterium, the same one that you're going to spend 4 months using to produce a stable plant. You can just test it really quickly. And I get results in like 3 days' time, right? So you inject a petal today, and then 3 days later, you have a signal. Again, this is just temporary to test all the stuff and to make sure that my plasmid isn't toxic, for example, right? Because like those petal cells are actually pretty fragile and they're still metabolically active. So if there's something wrong with the genes that I use, like I'm making too much of this one gene or something, it'll just brown out really quick. So it gives me a good indicator as to whether or not I want to commit the next 4 months to something that will probably kill the plants halfway through.
So, if it was a desirable change that you wanted to make permanent, since you said that agroinfiltration is not permanent, would you then use the gene gun to try to get that gene permanently growing in that plant and grow it out from tissue culture? Is that how the
Um,
process would look?
it depends on the plant, right? So if the transient infiltration works with Agrobacterium, then the stable transformation with Agrobacterium will also work. And so what I do is I take a bit of the plant that I injected, not the actual injection tissue, but just of leaves, and I co-culture. Surface sterilize the tissue. I make some leaf discs, and then I co-culture the leaf discs with the Agrobacterium that I used, right? In the same type of modality. So whenever I want to do an injection, I make 2 tubes. One that I'm going to inject, and another one is backup in case the injection works. And so if I'm really confident, I do both at the same time. But essentially, what I would do is I would grow up Agrobacterium, make it virulent, and then expose it to those leaf discs for about 20 minutes, and then put it on media in the dark such that the bacteria and the leaves co-culture on your plant media for 72 hours. So you're like purposely contaminating X plants with bacteria. And if you wait the 72 hours, it's around the time that it takes for Agrobacterium to transfer its DNA fully, right? Just like how we get signal in 3 days, similar process. And then you want to kill the Agrobacterium and keep the plants, right? And so you move it onto media that has antibiotics that kill the Agrobacterium, right? And so now you're starting to do tissue culture with plants where around the rim of the leaf discs, you have hopefully transformed cells, right? So transgenic cells, and then you do the process of tissue culture to regenerate those leaf discs into whole plants, with the added caveat of every media that you use from now on will also have the herbicide that you added on your plasmid.
Got it. Okay,
Yeah,
that makes sense.
yeah. So the only thing that will regenerate from your tissue culture will be plants that are transgenic.
So cool. That's so cool.
It's pretty accessible, especially if you have a tissue culture background. You start realizing that all you have to do is add one extra step and one extra thing to your MS media, right?
Yeah, I was looking through your protocol library because you have a lot of information available out there about how to actually work with the agrobacterium. So I was thinking it might take me a while, but over time I thought it would be a cool video to work on.
Yeah, absolutely. And I have a bunch of agrobacterium plasmids that I'm happy to share if you need any to just jumpstart your process.
I saw
Um,
them on your website. They look really cool. They almost look like Gatorade sorta. They're pretty.
yeah. So I have a bunch of chromic protein plasmids just so that people can do agar art with. They're like genes from coral that have different colors in daylight. And
Mhm.
then I have my ruby green plasmid, which makes green in agrobacterium and ruby in plants, right? And that allows you to like track the agrobacterium as it's doing its thing, and then you know it did its thing because then the plant is suddenly red, right? The one that you're generating.
Fascinating. How long do the plasmids store for?
So in agar stab forms, they actually store a really long time. Like the longest I've had was 5 years that I
Oh,
just pulled. Yeah, I had a tube in the back of a drawer and I said, okay, let's see what happens.
yeah.
I forgot about it. And then I poked it with an inoculation loop, struck it out on a plate, and it grew. And so if you don't open the tubes, they last for years. If you open the tubes, I think you can double dip maybe twice before the cells start like kind of getting damaged. But once they're on a petri dish, what you want to do, because I sell them in E. coli, not in agrobacterium. And so what you would have to do is this thing called a mini prep that basically you grow the E. coli in liquid culture, and then you purify the plasmid from inside the E. coli using chemicals. And I have a protocol for that. It's called the columnless mini prep. And it basically uses soap and sodium hydroxide and some sodium acetate to break open the cells and then precipitate all the E. coli guts, such that
Hmm.
the only thing that's left is those little circles of DNA, the plasmids, in liquid. And then from there, you would transform Agrobacterium
I
with
see.
it. Yeah. So the E. coli is more of like a storage vehicle. It's a construction and storage vehicle of genetic elements, like plasmids, that you then can extract the DNA from and put it on another organism.
Why is it always E. coli?
I think so. We were like building the plane while we were flying it, right? So the reason why E. coli is so popular is that it came from a diphtheria patient in the 1920s, right? And this doctor was doing some analysis, and they found that the microbe was non-pathogenic. It didn't have the mechanism of taking up residence in the human gut, right, to do all of its damagey stuff. And so they found the chihuahua of wolves in somebody's poop, right? And they're like, let's study the chihuahua to better understand the wolves,
Hmm.
right? Because working with a pathogenic microbe is dangerous. You need certain confinement. And back in the 20s, we definitely didn't have what is like a biosafety level 2 or 3 facility to keep everything as intrinsically safe as it is now.
Yeah.
And so they thought, let's work with the broken, dumb E. coli so that we can understand the really, really bad stuff that's killing people with cholera, right? Um, and so that became the most studied organism on the planet, right? Because it's like the first example of direct medical relevance, not just studying a microbe for the sake of it, that a lot of people were putting a lot of money into in terms of like funding for research. And as the story goes, we saw that there are microbes that have plasmids, little circles of DNA, naturally. And so we started studying the plasmids of microbes and using E. coli as a model for understanding other microbes. And what we noticed is that extracting DNA from it became really simple because people put a lot of time and effort to streamlining that. Because it's non-pathogenic, it can grow in fairly educational environments without much control. And slowly but surely, it became like the model, the lab rat of microbes. And so we have all of these protocols and all of these things in place to work with them. But now, with the advent of deeper sequencing, more affordable sequencing, better understanding of microbiology in general, there's a lot of research happening in non-model organisms, right? Things that haven't been elevated to model status in terms of depth of understanding and working with it. And now it's becoming a popular field. Like I see papers all the time of like a toolkit for engineering this bacteria or that bacteria, and even archaea, which are my favorite personally, where it's like kind of in a midway between a eukaryote and a prokaryote, this like secret third thing, literally. And so yeah, so there's a lot of tools now for you to work with it, but E. coli just is the tried and true reliable workhorse, and it works. More importantly, like
Yeah.
bioengineering at the end of the day is really pragmatic. We work with what works and don't fix what's not broken. And that leads to some dogma where like people would ask, why not something else? And they go, if you have time, go for it. But this works. We have
Yeah.
so many other bigger problems, right? Than to worry about like how to do cloning in a different microbe. And more importantly, a lot of the strains that we use for E. coli now have been further engineered and removed certain genes, like the genes for gobbling up DNA really quickly. They deleted those, so plasmids last a longer time inside those cells. Or if you want to produce protein, they deleted the genes for proteases that naturally gobble up and recycle proteins inside the cell, such that your proteins last longer, right? So you can extract and make more of it. So we've added a lot of quality of life upgrades to this microbe so that it's really worth just working with those.
Sebastian, thank you so much for answering all my questions that I had about genetic modification. I think you're really talented at explaining very complex topics to someone without a lot of background in the field, like myself. So I really
I appreciate that.
appreciate that. My last question for you is just if you had any advice for people who wanted to get involved in science, specifically plant science, who don't have a formal education or a STEM degree necessarily.
So I get this question a lot, and it's one of my favorites, and I answer it differently every time. I wish I had a better, more concise answer. But what I would recommend And this is so any advice that I give is always from a point of my own personal failure, right? So like I tell people not to do something because I've done it and it was awful, right? It's
Hmm.
not, and some mistakes you do have to just learn on your own. But looking back in hindsight, it took me a really long time because I didn't have two things: mentorship and a question, right? The mentorship part is really hard to come by if you're not in a university environment to begin with. So, like, my number one advice is stay in school. If you can have access, if you're at a university and there is a plant lab and you're interested in plant biology, just go up to the professor and be like, what do I have to do to work with you? Like, I'm happy to volunteer time or whatever. And now there's paid internships too, right? Which, like, getting involved in an academic environment is always one of the best ways to get involved, right? Like
Hmm.
local community colleges could have these courses that if you ace that course, maybe you can TA, right? Like there are ways to kind of ingratiate yourself, even though it's not like some fancy Ivy League. Good science happens everywhere. Now, in the uh, in the event where you don't have access to a university or a community lab, I haven't mentioned community labs, but real quick, they're kind of like gym memberships for biology, and they're popping up in all major cities where you pay a monthly fee and you have access to all of this smancy laboratory and like PhD level folks who are there, and there's projects that are already ongoing. So if you do join a community lab and you have no experience with science, definitely hop onto a community project, something that's already ongoing, and seeing where you can contribute so you can learn the ropes. Um, it's if you don't have access to any of those and you want to do things on your own, the first thing to do is to figure out where you are on this planet, right? So where you are determines the breadth and depth of how much you can do, right? So in the United States, the biosafety level 1 is not regulated, and so as a result, I can do what I'm doing. But in places like Europe, for example, it's like the equivalent of a federal felony to do what
Hmm.
I'm doing in an informal setting. And so avoid the genetic engineering, especially early on. And just my biggest recommendation is to do tissue culture, to learn plants through culturing them. And in tissue culture, because there's no regulations for this, outside of like growing like opium poppies or weed in certain countries, um, it really allows you to do two things. One is better understand the organism in a really isolated environment, right? Like Barbara McClintock, one of my heroes. She was such a badass. I wish I met her. She wrote this book and basically said she said you have to get to know the organism, develop a feeling for the organism. And I know that sounds kind of like hippy dippy and romantic, but I honestly cannot recommend it enough. If there's a plant that you want to get to know it, right? Like engage in tissue culture, learn about the literature, read up on the field in question of the plant that you're interested in. If you don't, you can just go into your backyard, pick up a random plant, and see if you can culture that. Right. Videos like yours are super helpful because it allows visual context that would otherwise just be in a book. So like I wish channels like yours existed when I first started, right? Because I was like reading this book and going like, okay, mix the media. I'm like, well, how?
It's
What does it look
hard
like?
from text. Yeah, I try to make videos that would have been helpful to me 2 years ago. That's my goal.
Yeah, yeah, and the like I'd love to do videos on like the genetic assembly and design aspects of it, so that you can physically see like how to put the liquid in the tube and put them in the PCR machine. Um, but yeah, getting back to the original question, the having if you want to go down the biology route and you want to just like be a contributing member of a biological research community, um, the most important thing is to have a question. Before you build a lab, a home lab, build your question. Figure out where on the tree of life you're most interested in, and start reading in the literature around that, right? Like we have things like Anna's Archive and Sci-Hub for articles. I'm a big proponent of just pirating the papers. I'm not going to pay $30 for something I paid with my taxes to be funded. No.
True.
Um, so yeah, so read up on the literature around the organism that you're interested, around the topic that you're interested in. If it's ecology, if it's microbiology, if it's zoology, and then from an ethical standpoint, the reasons why I brought up zoology is that I am deeply, deeply against experimenting with nociceptive creatures, so things that can feel pain. And so, like, unnecessary suffering is not a good idea. Life is all suffering, especially in nature. The last thing you want to do is do it for the sake of entertainment. It's ethically and morally wrong to do so. So, if you are interested in plants, lucky you, they don't feel pain and they are an interesting blend of living material. right? And so within the realm of plants, there are so many plants that have yet to be fully domesticated, that have yet to, I'm not even talking about agriculturally, I'm saying like tissue culture wise, that have yet to be put in culture. You can write a small paper on the tissue culture of this plant, see if there are protocols for it. If they're not, look at you. You just did something novel.
Yeah.
It's going to be hard, of course. And it's going to be important that you write things down. So like I use Notion as my open lab notebook, and it doesn't matter what device or thing you use. It could be paper, it could be whatever, but write what you do down like a journal, like a diary. Like today I did this and I did that. And you'll thank yourself, because there's so many times where 6-month older me is just screaming at the me now, calling me a dumbass for not writing this down, because I really, really needed that number, that value, that thing.
Yeah.
So it's not science until you write it down. That's what Adam Savage said. Highly recommend writing things down. And begin with that question: what interests you? Do a literature dive to better understand the current state of the art. And remember that as an amateur biologist, as an amateur plant biologist, as an amateur plant bioengineer, even if something has been done, novelty is not as important as your own personal enjoyment. Because at the end of the day, you don't have to contribute to science to do amateur biology. You can enjoy it. Like I'm very hedonic of my practice. Like it's pure hedonism. Like I do it because I love it. I enjoy doing this stuff. Like it makes me happy. And if it's been done to death and you just want to do it, just do it. Don't chase novelty. That's not super important. And there's a replication crisis in science where you publish a paper, but very few actually get a chance to replicate. So sometimes replicating a paper and seeing different results and trying to be carefully describing what you're observing is both formative as a scientist, but informative to the overall community. Because maybe you might be the first person to have ever replicated that experiment, right? But again, glory hounding is not important. Just chase what you're interested in, build the question first, and slowly build up what you need around a very specific goal, right? Like set a goal and then only buy equipment that will help you in that goal. I failed by being a generalist early on, and I bought a whole bunch of crap off eBay. Most of it was broken. I had no idea what I was doing. And I'm like, this is what scientists need, right? They need all these things. I had no idea, right? And I don't blame you if you did that mistake, or you're actively doing that mistake. It's more of, this is how we grow without mentorship. It's just like you realize at the end of the day that half the crap in my lab I don't need.
Yeah.
Right? So be question focused and build up from there. And you'll start noticing that there's overlapping. Eventually, you'll get to a point where certain protocols within a new paper are familiar to you, because you start seeing the general workflow of tissue culture, the general workflow of bioengineering in that sense.
No, I
Please.
get comments all the time that are like, the most challenging part about doing tissue culture is reading a protocol. But eventually, when you read so many, you kind of just need to skim it and you can really pull out exactly what you need, like the sterilization procedure and the media recipe.
And the rest is the kind of like flowery language to make sure that you're not ripping off the million of other people who've described the same thing, right?
And then there's never any photos because it costs extra for them to publish photos. So you just got to go from the text,
So
which
dumb.
is hard sometimes.
Yeah, but the cycle can stop with us, and that's the fun part. Is that you can then digest an article into its base components, put it very like visually interesting, but also visually digestible. Like you can just make a quick cookbook recipe of that thing, right? And that's why Plants from Test Tubes was so impactful for me, because it digested those articles into cookbook style recipes, right?
Yeah.
I'm just like, oh, 4 grams of that, 2 grams of that. Cool. I don't have to read like the full chemical name with all the commas and numbers of the thing that just says BAP, right? Like I don't need to know benzyl aminopurine and alpha and naphthalene acetic acid, all that stuff. You will eventually, and it'll become, like you said, it'll be second nature. But things are hard until they're easy, and the only way to get from hard to easy is with work. So like there's really no shortcut, because if you take something that's pre-digested, you then never develop the muscle memory to digest it yourself, right? And there's something so liberating in being able to just pick up an article and it speaks your language. Because the art of scientific article writing is its own kind of language, because the jargon, one word saves you 7 sentences more often than not. And that brevity is important. So definitely practice reading and like for the folks who are listening, if you listen to one of Laura's videos and you see the article that she's mentioning, read that article along with the video, right? Like, see the side-by-side digest of what she's talking about and what the article is, so you can get a better understanding of, oh, that's what they meant, right? Because that, oh, that's what they meant is like the heart of science. Like, trying to communicate all of this is the main point of science. It's not in the sciencing and the pipetting and stuff. It's the communication of knowledge, the curation and communication of knowledge, which is an art, and not everybody writes well. And there's also now AI where you can just copy the thing and say, what the hell does this mean? Translate this into as if I'm 5. And that's also been really, really helpful for some students. Like, I've sent them a couple articles and they're like, this jargon hell is a nightmare. I ran it through ChatGPT and they like mansplained it to me like I'm 5. And every single line was more clear than that article. So now we have tools of translating those two, which is also helpful.
Sebastian, thank you so much. I find your work very inspiring and super cool. You've dug so much deeper into biology than I ever have, but hopefully in 10 years, I'm doing similar work to what you're doing, because I just think it is the coolest thing ever. And like I said, very inspiring.
Thank you so much. And hopefully, if my work holds true, it'll take you far less time to get to the same place. So fingers crossed that I'm going to be trivial one day.
Fingers crossed. The lighting was just so good, and now it's so bad. Thank you again to Sebastian, and also thank you so much to you guys for watching my videos. If you're interested in supporting the channel and the educational content that I make, I do have merch for sale. I'm not wearing it right now. I'll put a picture of it up. It's really cool. And I think that's it. Okay. Bye. Bye, guys.